Dr. Chatterjee is also there. I think I saw him joining. No,
Okay.
but I don't see him anymore.
Good morning, sir. Good morning, sir.
Good morning.
So should we start then?
Yes. Suchishmita, you start the session.
Just 1 second. Let me start the recording. Shourav, we have started live streaming.
Yes, ma'am. Yes, ma'am.
Okay. Ask for consent, accept. Okay, just started, and here also it has started. Dr. Chatterjee is also there. I think I saw him join. Okay. Very good morning and warm welcome to our respected speakers, Professor Ghosh and Dr. Chatterjee, to our respected Principal Madam, to all dear participants, to the second session of the webinar series entitled Industrial Application and Sustainable Development of Microbiology. And this is Dr. Suchishmita Das from the Department of Microbiology and IQAC Coordinator of GHK Jain College. Today we have an eminent speaker with us, Professor Sanjay Ghosh from the Department of Biochemistry, University of Calcutta. He is an excellent teacher, a famous scientist, and he is our guardian who guides us in every step for smooth functioning of the UG Microbiology course on the University of Calcutta. Whenever we teachers come across any problem, he is there to help us. Thank you, Professor Ghosh, for all your support and guidance, and for gracing this webinar in spite of having such a busy schedule. We're all looking forward to hear from you, sir. Thank you all for your nice feedback and valuable comments for our first session, which was held on 24th of August. We are glad that you all liked the session and that it was very informative. I have two humble requests. Some of the microphones are on. Few humble requests. Please do not switch your camera and microphone if you can, and please do not share your screen during the presentation. And type your questions in the chat box of Google Meet and YouTube, and they will be read out to the speaker after the lecture session. And today's feedback form link will be provided in the chat box only at the end of the interactive session, and please submit it tonight by 11 p.m. And please, it's a request, do not ask for it before the interactive session. And also please remember that the certificate will be generated after submission of the final feedback form on the last session, which is tomorrow, 29th of August, tomorrow from 11:30 a.m. Or tomorrow's session will be from 11:30 a.m. So with this, I now request our respected Principal Madam to introduce our speaker, our respected speaker, Professor Ghosh. Over to you, ma'am.
Thank you, Dr. Dash. And now it's a very good morning to everyone. I'd like to offer my warm welcome to Dr. Sanjay Ghosh and Dr. Abhijit Chatterjee and all other participants to the second day of our online lecture series. Now it's really a great honor for me to introduce such a dynamic and enterprising academician like Dr. Sanjay Ghosh to today's webinar. Dr. Sanjay Ghosh is a professor in the Department of Biochemistry at the University of Calcutta and also the chairperson of the Undergraduate Board of Studies in Microbiology. He completed his graduation in chemistry from Scottish Church College in 1989, master degree and PhD in biochemistry from the University of Calcutta in 1991 and 1998 respectively. Dr. Ghosh was a postdoctoral research fellow and staff scientist at Cleveland Clinic Foundation, Ohio, USA from 1997 to 2000. Dr. Ghosh's areas of specialization include microbiology, cell biology, and especially nitric oxide synthase and structure and function and nitrosative stress response. He received the ISCA Young Scientist Award in 1995 during the 82nd a session of the Indian Science Congress Association, Calcutta. He was also awarded fellow membership by the International Society of Biotechnology in 2008. Over the course of his academic career, Dr. Ghosh has held several positions which include convener of Research Advisory Committee in Genetics at Calcutta University, Deputy Coordinator at Center for Advanced Studies in Biochemistry, Phase 2 Department of Biochemistry, Calcutta University, and expert member of the PhD Committee of Department of Biotechnology and Life Sciences, Jadavpur University. 14 researchers have been awarded PhD degree under his supervision, and 6 researchers are currently pursuing their MPhil and PhD degrees under his guidance. Dr. Ghosh has completed UGC minor and major research projects along with CSIR, DAE, DST, ICAR, UP, and DBT projects till date. He has 58 publications to his credit, some of which have been published in the Journal of Applied Bacteriology, the Journal of Biological Chemistry, Journal of Applied Microbiology UK, latest in Applied Microbiology UK, Journal of Industrial Microbiology and Biotechnology, and International Journal of Microbiology. Really, we are very fortunate to have such an eminent university professor as Dr. Ghosh. Dr. Ghosh, I'm just handing over this session to Dr. Ghosh. So over to you, sir.
Uh, Thank you very much, Dr. Singh and Dr. Das, for your kind introduction. And I'm really happy to present today's talk. And actually, this area, uh, I like to present this area because in our choice-based credit system, we have this particular area in our syllabus, and I believe that students who are presently actually receiving this course, they must have a choice that either they will go to research or they can opt the industry. So the industrial applications of microbiology and uh, sustainable development, which is the theme of today's uh, seminar. And here I would like to present the art of food preservation techniques, because if we want to learn the food preservation technique, first of all, we are basically microbiologists. So in the angle of microbiologist's view, what we find that the food microbiology and the preservation of food and cereals and other products, that is basically aseptic transfer, aseptic preservation, and all the different microbiological techniques which are involved in this preservation procedure. So first of all, here the Louis Pasteur is the father of microbiology, and we all know that without his experiment, initial experiment, it would not be possible actually to invent anything in microbiology. And here I would like to mention that initially in his childhood days, when he was in primary school, he was most ordinary student according to his academic performance. Actually, he was much more interested in fishing and also in sketching. And later, when he joined in school, even that time also, his performance was not that much satisfactory. But interestingly, actually, what Pasteur said that fortune favors the prepared mind. So in the field of observation, chance favors only the prepared mind. So prepared mind, that is very important. And when he worked in the field of organic chemistry, that time actually he first time crystallized tartaric acid, and with this tiny tartaric acid crystal, he first time discovered that it is the optical isomers, actually the mixture of racemic mixture of dextrorotatory and levorotatory mixture of D and L tartaric acid. And that has opened up a new field in the organic chemistry, particularly the molecular characterization of the crystal structure in the field of organic chemistry. So it is gradually, and here we know that Louis Pasteur, he is the father of microbiology, and he showed first time that he destroys or kills microorganisms, and his famous swan neck experiment we all know. And actually in 1854, when he started his studies in fermentation, he found that each of the fermentation is carried out by living microorganisms. So he used his research in fermentation to fight against disease of wine or souring of wine in 1865. He realized that these were caused by unwanted microorganisms that could be
Professor
destroyed.
Ghosh,
Yes.
Professor Ghosh, sorry to interrupt. Can you make it that slide presentation, the slideshow or full screen?
Okay, sure.
Yes. Thank you, sir. Thank you. Sorry for
Oh,
interrupting.
no, it's okay. Okay. Is it visible now? Okay.
Yes, sir. Yes, sir.
Okay. So first time he realized that these unwanted microorganisms that could be destroyed by heating wine and a temperature between 60 degree and 100 degree. And so this actually theory was then used in all types of perishable items, including milk. I will discuss the pasteurization and the process of preservation of milk later. But here, first of all, we have to think about the conditions for spoilage. So here I'm showing that the foods that spoils quickly and foods that resist spoilage. So we all know that moist food like milk or any other juicy materials, natural foods, fish, banana, unrefrigerated food, yogurt, meat, ground or sliced meat, and those are easily spoiled quickly. And food that resist spoilage, so the dry foods, acidic foods, refrigerated foods, and whole meat. And the factors influencing the spoilage of food, that is the water, availability of water, pH, physical structure, oxygen, and temperature. And we all know that microorganisms cannot access their food material without the aqueous media. So all the foods are very good growth media for the microorganism. And the microorganisms present in the raw food material that can survive in the food and that can multiply in the food. So the controlling factors, which are some extrinsic factor, so the temperature, relative humidity, gases, contaminating microorganism, and the change in extrinsic factor that may lead to the enhancement of the microbial population, and intrinsic material, the composition and the physical and biological state, and change in this intrinsic factor that can also change the microbial community. So in this way, microorganisms can grow in food material. So food preservation, before going to start food preservation, why do we need food preservation? Because United Nations has committed to carry out zero hunger by 2030. So that means the zero hunger, we have enormous food material in our earth. We can produce many food material, but food preservation is important because it is not that all types of foods are produced in all parts of the world, and it is also important that some foods are produced in a particular season, not that for all season. And if we could preserve the food material, and we can preserve the food material so that we can distribute among the population. So food preservation is a process that food and vegetables are prevented from getting spoiled. The color, taste, nutritive values of food is also preserved, and food products lasts for a long period of time. Shelf life of food products are increased. And in this respect, that we know that retaining food over a period of time without being contaminated by pathogenic organism or chemicals. That is also important, and it also involves actually preventing the growth of the bacteria, fungi, and other microorganisms, as well as retarding the oxidation of fats, which can cause rancidity. And in this respect, I would like to say that the food preservation methods, that is a very ancient procedure, and this ancient procedure was used by the all we find that the mummies and the other preservation methods by the Romans, they describe that actually the they dries the food, enhance the flavor, beef, ham, bacon, fish, and they added formaldehyde, and smoking the food preserve materials, by which smoked material that can be preserved for a long time. And also the drying or desiccation or dehydration by sun, wind, or ovens, meats and fruit can be preserved. And salting, day salting and pickling, rock salt, sea salt, spice salt, cheese, olives, hams, eggs. Those are the early preservation method by mixing with salt. And where else has salting been used? And when just if we think about the preservation of mummy, so in that preservation method also different salts were used. So if we think about the food preservation as a whole, then food, we have to divide the food types into three different categories. One, obviously, the perishable food. Second category, that is the semi-perishable food. And the third category, that is the shelf-stable food. So perishable food items like the fruits and milks, semi-perishable like cheese, some ready foods, and shelf-stable, that is the grains. So some perishable food items, it lasts for less than 2 days to 1 week, like fruits, milk, vegetables, meat. And semi-perishable food items, it lasts around 2 months where under processed, like ice cream, cheese, bread, cake, pastries. And shelf-stable, it has longer shelf life, more than 6 months. That is, as for example, the food grains. So when we try to preserve the food material, the first of all, as I mentioned in my beginning of my lecture, that the aseptic process, to keep out of microorganism asepsis, that is very important. That when we are preserving the food materials, that time it should be preserved under aseptic condition. I can give you one example, like when you buy any baby food, what you will check first? Obviously, the date, date of manufacturing, and in the food items, sometimes it is mentioned that it should be used within 2 years after packaging or this kind of thing. But one thing is very important, that you should check the seal of the baby food, whether it is properly sealed or not. If it is properly sealed, if the container is not opened, then if it is within the dates of the expiry of the starting from the manufacturing date, it can be used. But if the seal is ruptured, then obviously you should not use that. Why? Because we find that in baby food or in any nutraceuticals or Horlicks or Compliance or any other branded the health food materials, actually you will find that if it is stored, even if it is closed, if it is not sealed, you will see that a layer of growth just on the top of the uh, that particular milk material, milk powder material, or baby food material, or in any maltose type of material. So these are the microorganisms that comes from the environment. And these microorganisms are mostly fungal spores. They can germinate on the top of the baby food material, milk products, even under dry condition. So why do they grow under that condition? Because the fungal cells, they require very low water activity. We know that in microbiology, we frequently use the term water activity. That means the available water that is free to react. And in bacteria, the available water free to react, that is zero point 0.9 to 0.95, and there is a relationship between this water activity and relative humidity. So if the relative humidity is 95%, that means the water activity is 0.95. So in case of bacteria, we find that bacteria, they require more water activity. And in case of fungal cells, the water activity requirement is much less, that is 0.5 to zero point 0.6. Within that low water activity, even they can grow. So in a solid support food material, fungal cells still can survive in that low water activity. So that is the aseptic condition. Maintaining the aseptic condition, the food material, that is very important. And the second part, that is the removal of the microorganism. That means by stopping the growth and activity of the microorganism. So we know that the control of microorganism from the food material, basically by means of physical agent or chemical agent, and we are basically dependent on low temperature drying, addition of chemicals, and another method. So low temperature drying chemicals that cannot ensure that all microorganisms are destroyed. So we can call them a microbiostatic conditions. But destruction of microorganism by heating or radiation, that is the microbicidal condition. Because in this procedure, we ensure the killing of microorganism, even its endospores. So by means of heating and radiation, we want to kill all types of vegetative cells, including their endospores. So protecting against self-decomposition of food, that is also very important, because endogenous enzyme, phenolase enzyme, in plant products, there are lots of polyphenols. So these phenolase act on this polyphenolic compound to produce melanin, and that can turn the vegetable into brown in color. So this enzymatic browning, and there is also non-enzymatic browning. So if it doesn't look good, the food material, so people would not like to take that particular vegetable if it doesn't look good. So obviously the fresh vegetables, if you want to preserve the fresh vegetable, you have to inhibit the activity of the endogenous enzyme, that is the phenolase enzyme. And also you need to delay or inhibit chemical reactions, non-enzymatic browning, and protection from invasion and spoilage by insects or rodents, protection against losses of mechanical causes. So you know that we have many traditional techniques. Always I say that our great grandmothers, actually they knew that home science, that they were not microbiologists, but they knew that how to preserve the food materials, because they knew that how to preserve the pickles, and they knew that if the pickles are placed in sunlight, so they are not spoiled by the fungal cells. And they knew that the if the materials, if the materials need to be preserved, so the clothes that must be freshly washed clothes. So they knew that the if they do not don't wear freshly washed clothes, so that can spoil the food materials. So that means the from clothings something may come into the food materials, so that can spoil the food also. So these are the pros and cons of this food preservation. Actually, they knew, and these are the traditional techniques. So, and also they knew that if the dried neem leaves that can be placed in cereals, in rice, in wheat, if those are placed, then the pest can be controlled. They knew that actually the red chili, dried red chili, if those are placed in food materials or the grains, food grains, so pest can be controlled. So protection against losses by mechanical causes, I will come to in that part later. But basically what we have learned that preservation methods vary according to the food items and quantity of the items to be preserved, and principle of food preservation can be broadly classified into two types: by means of bactericidal methods and by means of bacteriostatic methods. Uh, so up to this point, everything is fine. Do you have any questions?
I mean, sir, there are questions. Should we read to you
Okay.
now or discuss them later, right?
After finishing the lecture, then we can
Yeah,
address
yeah,
the
yes,
question,
yes,
right?
yes, yes.
Okay.
Not finished.
Okay. Okay. So up to this point, is it okay?
Yes, sir.
Yes, sir,
Okay.
absolutely, absolutely, sir.
Okay. Okay. So we know that bactericidal methods, most of the microorganisms are killed by cooking, canning, pasteurization, sterilization, irradiation, etc. So the bactericidal methods, in India, we love full cooked material. So that is why we don't need to think about botulism, because the food-borne pathogens, Clostridium botulinum, it can produce a toxin that is botulinum toxin, and that can cause the spastic paralysis and neurological disorders. So these kind of actually it can stop the neuronal signaling by means of producing these toxins. But these Clostridium botulinum spores are usually associated with fish material. So the canned fish material, if that even after sterilization, if the Clostridium botulinum, if that can escape the sterilization procedure, so then the botulinum, Clostridium botulinum spores can be present in canned food material. So that if it is half cooked and the spores are not destroyed, so they get sufficient time to produce the vegetative cell and to produce the toxin. So in India, we don't need to think about the half-cooked material, because the cooking procedure, even using the pressure cooker, that is a very good method because it can kill all the microorganisms. And also by means of pasteurization, it doesn't mean the sterilization, because pasteurization does not ensure the full destruction of the microorganism, because we know that pasteurization is the process of heating every particle of milk or milk products to at least 145 degree Fahrenheit and holding it continuously at or above this temperature for at least 30 minutes, or at least 161 degree Fahrenheit and holding it continuously at or above this temperature for at least 15 seconds. So one is for long time, that is for 30 minutes. One is for short time, that is 15 seconds. So the 161 degree Fahrenheit high temperature short time pasteurization, that is also very useful method. And nowadays, actually, you will find another milk, that is Amul Taja or that kind of milk. So that is ready to eat. So you don't need to boil the milk. And actually, in this procedure, they expose milk to ultra high temperature for a very short period of time, 300 degree Fahrenheit or 148.9 degree centigrade for 1 or 2 seconds. So the final product is compared in flavor and nutritional quality to pasteurized milk. So it does not require refrigeration, and it has indefinite shelf life. But still, we don't like to preserve the milk material at when it is sterilized at ultra high temperature for a long time. Within 1 month or 2 months, we should take that milk material. So if it is preserved, even the shelf life is very high. So it doesn't need refrigeration or low temperature preservation. And bacteriostatic methods may be achieved by removal of water, use of acids, oils or spices, keeping the foodstuff in low temperature, drying, freezing, pickling, salting and smoking. So physical methods that involves freezing and canning, usually drying, gamma irradiation, exposure to ultraviolet or high intensity white light, ultra high pressure and filtration. These physical methods, sometimes we know that depending upon the taste of the food. Suppose if you preserve the freshly prepared sweets in refrigerator, the taste is changed. So usually the freshly available sweet materials, if it is preserved in freezing condition, that may change the taste. And sometimes the excess sweet materials are stored in shop, not in refrigerator, usually under desiccating condition. So if the air is removed using a small pump, so under desiccating condition, the it ensures the very low humidity or very low moisture content inside the area, and under that condition microorganism cannot multiply. So under that condition sweet materials can be preserved. And but this canning industry is largely dependent on the gamma irradiation to ensure that even a single endospore can be destroyed. So lowering the temperature of food, uh, so that the basic principle, by lowering the temperature of food and by means of food, this technique, food can be preserved because the microorganisms which are present in the raw food material or the surface associated food material. Here we have to remember that microorganisms may present in the surface of the raw material, not in the deeper tissue. In case of fresh vegetables or even fresh meat or even fresh fish, any kind of material, in the surface microorganisms may be present. So by lowering the temperature, we can just minimize the metabolic activity of those microbes, and obviously at low temperature only the psychrophiles can survive and psychrophilic enzymes may become active, but the all the mesophilic microorganisms, their enzymes are inactivated at that low temperature, and moisture is changed to ice and microbes become inactive without water. And packaging of food maintains the color, flavor, texture that is important, and fast freezing helps maintain the nutritive value and the texture of food. So usually this kind of freezers are used normally to keep the frozen materials. We all know, we have seen in all the departmental stores, the frozen materials are preserved in that, in like that manner. So quick and fast freezing and slow freezing very important, because nowadays we used to buy all the frozen chicken materials, chicken nuggets, chicken popcorns, and these kind of materials. So after getting the frozen materials, if we preserve the frozen materials say for 1 year, it would be fine. But if we open the sealed bag and then if we thaw and again if we freeze the material, So by that slow freezing and thawing, that can allow the damage of the food cells. Ice crystals are big, so damage the food cells, causing loss of texture, nutrient, color, etc. As well as that can encourage the growth or multiplication of the microorganism also. So quick or fast freezing occurs at minus 25 degrees centigrade or less. So ice crystals are small and that do not damage the food cells. So freezing, there are several advantages of freezing. It is simple, safe, suitable for lots of different foods. It is best method for maintaining color, texture, flavor, nutritive value. Leftovers can be frozen. It can prevent waste, and bulk cooking and freezing save time and fuel, and frozen food is handy for emergencies. So there are a lot of advantages, but there are some disadvantages also. Cost of freezer and running cost, space needed for freezer. It must follow rules for freezing and thawing for food safety, and defrosting freezer takes time and effort, some damage to texture of food. And another important thing, when the frozen materials are packed, so the properties of the packing material, it should be suitable for the preservation. It should be strong, it should be waterproof, vapor-proof, grease-proof, easy to use, and sealable. So the strong polythene freezer bags, aluminum containers, aluminum foil, freezer papers, wax cartons, plastic boxes with tight lids, these are important for food preservation. And we know that the frozen foods like chicken can be stored for 12 months, frozen material, vegetables also for 12 months, beef for 12 months, white fish that is for 6 months, cooked pastry, lamb, oily fish for 4 months, soup sauces for 3 months, and minced meat. So the chunk of meat can be preserved for a long time, but minced meat, it cannot be preserved for a long time. And bread. So usually we know that if the materials are kept at low temperature, and if we unopen the container, then it is fine. But if it is frozen, if we freeze and thaw several times, and we can, if we look at the texture of the material, if we smell the material, we can find that the tastes and smells are not good. And even when the frozen materials, we made several aliquots of our cooked materials also, and we take one aliquot from the freezer, and when we warm it, we should consume it, the whole material. But if we warm it several times and if we do not consume the material, that may cause the propagation of the microorganism. And sometimes we find the slimy appearance on the top of the cooked food materials, because the uh, microorganisms associated in the food materials that can produce slime or exopolysaccharide, so that can give that kind of texture, slimy materials. And sometimes microorganisms can produce the toxins also in the frozen, in thawed material, as well as they can produce some pigment, and that pigment may change the color of the food materials also. So by if we want to use the our cooked material for several days, we must aliquot it. So that is the principle of food preservation, cooked food material. So once one aliquot is taken, it should be consumed, and it should not be heated for several times. So by means of freezing and heating, and by the in that manner, that can encourage the multiplication of the microorganism. And another very important procedure, that is the blanching vegetables. So blanching vegetables, why blanching is required? Because actually to inhibit the enzymatic activity which is present in the vegetables, several phenolases, if the blanching is done, the enzymatic activity is also inhibited, and the color texture, it looks like the like very fresh materials. So the equipment is a large saucepan and wire basket or colander or large bowl. So prepare 5 liters of boiling salted water, prepare and chop vegetables. So blanch 500-gram vegetables at a time, and let's say time should be fixed, time from when water reboils, carrots for 4 minutes, and after, or similar, like you have to standardize the blanching vegetables, how long it should be boiled, and then plunged into ice cold water for same time as for blanching, and then drain well and freeze. So usually this kind of blanching procedure that may give the freezer burn if protein food is not wrapped properly in the cold air, will cause the food to toughen, discolor and dry out, and this is called freezer burn. So now another important point, nowadays we are very much skeptic about the preservatives, because most of the ready food materials that contains preservative, and in this preservative, like some preservatives are not that much toxic, but if we consume much preservative, that may become potential carcinogen also. So preservatives work either as microbial poison or reducing the pH to a level that prevent the growth of the microorganism. And chemicals used today are nitrates and nitrites to preserve meats, sulfides to prevent browning of fruits and vegetables, to prevent the fungal spoilage, and a list of preservative, although it's a very it's not very legible, but I just want to mention the list of preservatives, like the sodium propionates, sorbates, sodium benzoate, parabens, sulfides, as well as ethylene or propylene oxides, sodium diacetate, dehydroascorbic acid, and also sodium nitrite, caprylic acid, ethyl formate, and when the ascorbic materials, so that is not harmful. So usually it is a very good antioxidant materials. So any kind of reactive species that is generated within the chemical reaction during the preservation of the food materials, that can be scavenged by the ascorbate. So the other types of food preservatives which are used sometimes, particularly we know that and nitrates that are not good for human health. So the frozen meat, that is not encourageable, because the preservatives which are used to freeze the meat materials, usually the nitrite or nitrate. So can cause nitrosative stress, also oxidative stress. So today we use some of the methods for industrial food canning, and here I would like to mention that actually this canning industry, it's an ancient procedure. In 1810, Nicolas Appert, a Frenchman, described his successful research in food preservation. In the same year, Peter Durand and was granted an English patent, and for describing the use of containers, tin containers, for food preservation. So 1810, long time back in the history of food preservation, then Nicholas Appert and Peter Durand, first time they described the canning procedure of the food preservation. So in commercial sterilization, steam under pressure or retort, minimum temperature to reduce the Clostridium botulinum, and thermal death point that has been decided. So it reduces should be the 12 logarithmic scale. In that way, the finally it should be 10 to the power 12 logarithmic scale reduction, and finally the canning industry actually, or the bottling industry, that is much more dependent on this type of food preservation techniques. But commercial preservation, commercial sterilization, we have to ensure that endospores of all thermophils can be destroyed. But we know that thermophils can survive in commercial sterilization also. And sometimes Clostridium botulinum also may escape the commercial sterilization procedure. And that is why from the canned food, the botulism may occur. And thermophils, they may survive even greater than 45 degrees centigrade storage, and they can produce gas, soiling, and mesophilic bacteria can also survive if it is improper heating is done. Acidic foods or 100 degrees centigrade survivors, sometimes they can't stand the low pH. So these kind of there are several factors that can influence the commercial sterilization procedure. So washing, blanching, and filling, and steaming to exhaust air and sealed, and finally the sterilization and cooled, and it is level stored and delivered. So radiation is much more widely used technique for canning industry, which can escape by means of the heat sterilization or by those materials which are not heat stable materials, and heat may change the food nutrients as well as the texture as well as taste. So ultraviolet radiation, we all know that the UV radiation, the penetration power of UV is very low. So only UV can UV can penetrate the surface of the food, not in the deeper part of the food materials. So gamma irradiation, actually it can use the ionizing radiation. So it is also called the cold sterilization procedure, because a very low amount of heat is generated during the sterilization procedure. And using this gamma irradiation, it is possible to increase the shelf life or sterilize meats, seafood, fruits, and vegetables. So gamma irradiation, it kills insect and parasitic worms also, prevents sprouting of fruits and vegetables. So this is the schematic presentation where the actually in conveyor belts, the all the materials which needs to be gamma irradiated, and that cobalt 60 proper shielding must be ensured in this gamma irradiation procedure. So materials to be irradiated and irradiation source lifted from storage pool for processing period. So you can see the direction of conveyor to move materials in and out in the processing position. And irradiation treated foods nowadays: wheat flour, wheat, white potatoes, pork, fruit, vegetables, spices, vegetable seasonings, poultry meat, and frozen meat. All these are sterilized by means of gamma irradiation. Another very important food preservation procedure is pickling, because pickling food in vinegar or in acids make it difficult for microorganisms to live. And in our daily life, we found that the chili, green chili, can be preserved at low temperature. But at room temperature, if you want to preserve green chili, if you put green chili in vinegar, so it can be stored for a long time. And commonly pickled food include onion and soybeans and chilies that can be pickled and that can be preserved. Sugar can also be used in pickling fruits such as nutmegs, mangoes, and cherries. So basically, the sugar or brine that can exert the osmotic pressure on the associated microorganism which are present associated with the food material. So the it can cause the plasmolysis. And in that way, concentrated sugar solution, even in uh, India, you will find although we have lot of fresh fruit material, but the canned fruit like the pineapple, mango, that can be preserved in concentrated sugar solution, and that actually draws water from the fruit, and that can prevent the growth of the microorganism. So another very important area, that is the pulsed electric field. So in this, what is pulsed electric field? And a new area where the pulsed electric field is used for short electric pulse to preserve the food. And pulsed electric field treatment is an innovative and promising method for non-thermal processing of foodstuff. So the materials which cannot be sterilized by means of heat or by gamma irradiation, pulsed electric field can be applied. It is one of the most appealing technology due to short treatment time, typically below 1 second, reduced heating effect, reduced energy lost during the heating food is minimized for fresh like characteristic of food along with high sensorial quality and nutrient content. It is suitable for preserving liquid and semi-liquid food, removing microorganisms, as for example milk, fruit juice, soup, etc. So the basic principle is to keeping food below temperature normally used in thermal processing, but PEF technology is the application of short pulse of high electric field with duration of microsecond, micro to millisecond, and intensity in the order of 10 to 80 kilovolt per centimeter. So this processing time is calculated by multiplying the number of pulses times with effective pulse duration. The process is based on pulse electric currents delivered to a product placed between a set of electrodes, and the distance between the electrodes is termed as the treatment gap of the PEF chambers. So the applied high voltage results in electric field that causes the microbial inactivation. So it is the schematic presentation. In this schematic presentation, you will see that the raw product is pumped in through temperature chamber. So the treatment chamber, actually high voltage pulse is generated, and in that finally the treated product is collected through cooling coil, and the product is finally collected. So this is the commercial 25 kilovolt or 25 kilovolt pulse electric field PEF technology pilot plant capable of up to 500 liter per hour high throughput. So nowadays, all the juice processing, that is actually dependent on PEF treatment. So when the PEF treatment is introduced, juice of exceptional sensorial quality is obtained that closely resembles the juice of freshly squeezed fruits. And it is also safe in terms of microbial point of view because the chemical preservatives are not added in this. So as the chemical preservatives are not added and still the shelf life is increased, so additional advantage. And so this extension considerably, it can simplify this test of juice that otherwise would have expired. So as it is increasing the shelf life. So beyond pasteurization, nowadays another milk processing procedure, the smart milk project, that will develop a new milk treatment method by merging the thermosonication and pulse electric field. So in this technology, previous research indicates that milk treated this way retains more of its natural taste and nutritional characteristic than pasteurized milk. So while still being very safe to drink. Now I am coming to my last part of my talk. So we have discussed many techniques. Basically, most of the techniques are either microbiostatic or it can ensure the microbicidal technique. So still, major foodborne infectious diseases are very common that is salmonellosis, which I can mention. Even salmonella can survive under freezing condition. Under cycrophilic condition, even salmonella can survive. And that is why ice cream, if it is too old, just look at the dates. You should not take an old dated ice cream. You should not buy that ice cream. You should not consume that because it can cause typhoid. Because if anyway Salmonella typhimurium, it comes and inside the food materials, and when it is processed, the ice cream is processed, and if it is thawed several times, you never know the freezer if it is turned on and off several times. So looking at this date, it is old. So even in departmental shop, you may find that 50% discount on food item, 75% discount on food item. You should not buy those food items because the departmental shops are provoking you actually to buy those things which are not actually good for your health. Another thing, that is the Campylobacter or Arthobacter. I would like to mention the Campylobacter jejuni. Campylobacter jejuni, it can cause a symptom within actually 8 to 10 hours. You will feel the symptom, abdominal pain, severe abdominal pain, sometimes in the right side of the abdomen. So usually doctors cannot diagnose campylobacteriosis or the abdominal appendicitis unless the USG report is in doctor's hand, because Campylobacter jejuni, that can infect poultry chicken. So when the chicken is cut in a cutting board, and the same board is used in using the salad items also, and after, if the chicken is infected with Campylobacter and again it is frozen, so the Campylobacter jejuni, that may present in that actually frozen food material. So frozen chicken, Campylobacter may be present. So when it is thawed and when you are dressing material, so somehow Campylobacter may come into the food material and it can survive. So most toxins, and when it survives, it can produce toxins, and toxins are heat labile toxin, but varying periods of times, it may be some toxins are heat stable toxin also. And as I was mentioning regarding the salad preparation, don't take salad outside your home, because you never know these when salad is cut in a cutting board and it is properly washed or not. If the chicken is cut in the same salad cutting board, then directly Campylobacter jejuni may come into the salad material and you can directly consume the Campylobacter jejuni through your salad material. So another important disease is listeriosis, and caused by Listeria monocytogenes, and related to meningitis and abortion, newborn, elderly, especially susceptible. It is also associated with meat products, especially pork and milk. Escherichia coli, it can cause diarrhea and colitis, so an H organism cannot survive. Usually when we buy egg, we properly wash egg with water and then we put it in freezer, because the raw egg material, the eggshell associated environmental material, feces material, that may contain several microorganisms, and that may multiply inside your freezer along with other materials. And also Yersinia enterocolitica, that is associated with milk or meat products, associated with tofu, and also the several other Vibrio species like the Parahemolyticus, that is associated with seafood or shellfish, that can also cause intoxication. And intoxication of toxins in foods in which microbes have grown and include staphylococcal food poisoning, botulism, which I have previously mentioned, Clostridium perfringens, Bacillus cereus for food poisoning. And the toxin associated, like the ergotism, toxin condition caused by growth of the fungus in grains, aflatoxin, the carcinogens, aspergillus flavus produce the aflatoxin, and fungus infected grains and nut products, and fumonicins, carcinogens produced by fungus infected corn. And nowadays, several detection methods have been developed. But one thing I should mention, that detection of foodborne pathogen, it must be rapid and sensitive. And the methods include culture-dependent methods like culture techniques, maybe too slow sometimes, immunological techniques, very sensitive, and molecular biology techniques, probes used to detect specific DNA and RNA of the microorganism, and it is also sensitive and specific. So we can summarize this like to detect the foodborne pathogen, obtain the microbial sample, identify the pathogenic strain depending upon the culture-based method or immunoassay or PCR-based method, or by means of biosensors using freight or aptamers or gold or silver nanoparticles, or these PCR-based techniques, quantitative PCR, real-time PCR, and race PCR and multiplex PCR, all these methods can be used to identify the trace amount of microorganism associated with food materials. So at this end, I have discussed many techniques. Also, I have discussed the microorganisms which are associated with these food materials, and thank you for your patience.
Thank you, sir, for such an informative and a lucid presentation. It will be really very much beneficial for the students because it is in their syllabus, and of course for our teachers also, because we got to know so much from you, sir. There are plenty of questions are there in the chat box, so we'll now move on to the interactive session. And this session coordinators are Dr. Anamika Ghotok and Dr. Shonima Talabatu. Over to you all. Thank you, sir.
We find many fungal infections, not any bacterial infections.
Okay, thank you, sir. The next question is from Madhuri Jain. How does water activity affects lipid peroxidation in food?
Okay, very good question. Lipid peroxidation and water activity. Obviously, as I mentioned, when you dry the food materials, then the dry food materials water activity, you are actually dehydrating the water molecule from your system. So in that case, the obviously lipid peroxidation will be much less. And as the materials are dehydrated in absence of the water molecules, the lipid peroxidation rate will also be low.
Okay. There are two questions from Joita Khara. First one is, in mummification, which kinds of salts are mostly used?
Okay, good question. Actually, in mummification, it was observed in National Geographic Channel. If you follow, then you will find several YouTube videos are available how the mummies were prepared. And in mummification, mostly nitrite and nitrate salts were used to preserve the human flesh materials.
Her second question, can botulinum toxin be destroyed by heat?
botulinum toxin can be destroyed by heat if it is full cooked material. Usually, if it is 100 degrees centigrade, or if we boil the material, then obviously toxin is inactivated. But sometimes botulinum toxin is much more effective. Like, I can give you one example. If you go to Esplanade area, you will find uh, several restaurants. Actually, they're preparing their food material in one side of their cooking material, that is the half cooked chicken, and in other side, that is half cooked gravy. So if you order anything, then they will prepare the material half cooked chicken and half cooked gravy. But this half cooked chicken and half cooked gravy, when it is mixed, if that chicken is contaminated with Clostridium botulinum, at that temperature, Clostridium botulinum spore, they get sufficient condition for germination. So when it germinates, it can produce botulinum toxin. And when you take your food, you are directly ingesting the botulinum toxin along with your food material. But not that I am discouraging to take food material from outside, but always I would like to encourage the full cooked material, particularly in tropical countries like in India.
Okay. The next question is from Ognishuddha Shaw. He is asking from blanching part. Does blanching reduce the nutritional value of veggies?
Usually, blanching preserve the nutritional value, but definitely the if you compare the fresh vegetable and blanching vegetable, definitely you will find the difference of the nutritional value. But the frozen materials, the advantage of the frozen materials, sometimes it is easy to carry, or in the frozen containers, or that particular material is not available in that area, so people are compelled to take that kind of frozen material. But definitely nutritional quality, if you compare the fresh vegetables, it must be greater than the blanched vegetables.
Okay. Vidhi Modi is asking, does adding preservatives or additives in food lower the nutritional value of food in any way?
Actually, addition of preservative, it doesn't affect much the nutritional quality, because there is restriction and there is recommended dose for the preservative. And if the recommended dose is maintained, that preservative, that is enough to inhibit the growth of the microorganism. So it doesn't interfere with the nutritional quality much, but too much consumption of the preservative is not good for health. If you take the preservative containing materials daily, that is not good for your health.
Next question is from Shubham Bishas. What can be the disadvantages of consuming chemically preserved food materials?
Obviously, there are several disadvantages. The first of, obviously, as you are taking the chemicals with your food materials, and that chemical may disturb your metabolic activity, and that chemical may be carcinogenic, and that chemical is not good for your health. But it depends upon that, actually, how frequently you are taking that frozen materials or preservative containing materials. Like, I want to mention, like, that monosodium glutamate, Ajinomoto, if all the hotels, they stop using monosodium glutamate, you will not buy their food materials, because monosodium glutamate enhance the taste, and all these type of Mughlai food materials that contains monosodium glutamate. So if you regularly consume these kind of materials. So you are consuming monosodium glutamate regularly, and monosodium glutamate, it can cause cancer also.
Okay. Next question is from Riddhi Chatterjee. Will the consumption of frozen or preserved meat cause any diseases or any long-term effects?
Definitely, it can cause diseases and long-term effect. And as preserved meat material contain nitrite and nitrate, so when you are using dietary nitrate, dietary nitrite or nitrate. So these dietary nitrite and nitrate may produce several reactive nitrogen species. And these reactive, along with this reactive peroxynitrite is very important, which can cause post-translational modification of protein nitration. And these dietary nitrites can cause can produce several reactive nitrogen species, which can inhibit the mitochondria as well as mitochondrial biogenesis, mitochondrial ATP production, as well as several other targets. It can cause lipid peroxidation, it can cause protein oxidation, it can cause DNA oxidation. So long-term preservation of nitrite and nitrate is not good.
Okay, sir. Thank you for your nice explanations. The next part of the interactive session will be conducted by my colleague, Dr. Shonima Tolapatra Ghosh. Over to Shonima.
Thank you, Anamika Di. And thank you, sir. There is a second question from Riddhi Chatterjee, that how Clostridium botulinum is killed during food preservation.
Usually, Clostridium, as I mentioned, that canned food material, the canning industry is dependent on gamma irradiation. So, gamma irradiation that can ensure the destruction of the endospore of Clostridium botulinum, but still there are certain Clostridium botulinum strains which are resistant to gamma irradiation. So if that particular strain are resistant to gamma irradiation, that can be present in the canned food material.
Thank you, sir. Next question is from Paroma Dasgupta. She is asking, nowadays some milk products are available which do not require refrigeration and that can be kept at room temperature for 6 to 9 months long. So what is the principle behind this type of preservation?
Okay, when I was discussing in my talk that actually nowadays another preservation technique, that is the ultra high temperature. Ultra high temperature means that is 300 degree Fahrenheit or 148.9 degrees centigrade for 1 or 2 seconds. And in this procedure, actually they ensure the complete destruction of the microorganism. As there is no microorganism present in the milk material, it can be preserved at room temperature. So there is no question of multiplication of microorganism at room temperature. But if the bottle is opened, that should be consumed quickly. But if it is unopened, the ultra high temperature treated milk, and that milk actually the nutritional quality is also compared with the pasteurized milk, and it doesn't require refrigeration. And it is found that like Amul Taza or many other companies, they are selling this ultra high temperature treated milk. And also I mentioned about the pulse filled PEF technology system, and that using that technology also nowadays it is possible to prepare the increased shelf life containing food materials.
Okay, sir. Dr. Gargi Shaha Kesh is asking, under this current pandemic situation, how can we be sure about the preserved and packaged foods like biscuit, bread, etc.? I mean, is there any chance of them getting contaminated with SARS-CoV-2?
Very interesting question. Actually, what I believe that microorganism and virus, the surface associated virus, this is actually the inert material for the virus. So If it is properly packed biscuit or any other food material, when you open the material and you are placing the material in a good container, that is properly closed container, so you can take that, you can use that material. You don't need to take extra precaution, because now we are much more paranoid regarding the preserving, sanitizing vegetables, sanitizing every materials. But I do not believe, that is my personal belief, that we really require that kind of sanitization procedure. There is no report that SARS-CoV-2 can be present or can multiply upon vegetables. And even the vegetables, if a very small number of particle that is present in the vegetable, even after cooking, so after 2 or 3 days, if it is cooked or if it is cooked, there is no question of getting that SARS-CoV-2 along with the food materials. And the packed material, every time you do not need to sanitize the packed material. And sometimes it is advised, just uh, place the material in a corner of your home for 1 or 2 days. So after that, use it. If that satisfies you, you can use that. Otherwise, there is no chance of getting contaminated with SARS-CoV-2 with the food material.
Okay, thank you, sir. There is another question that which method is the best one to treat endospore forming bacteria, as they are UV and dry heat resistant?
Uh, Definitely, endospore killing. Moist heat are effective than the dry heat, and we use sterilization procedure 121.7 degrees centigrade and 15 pounds per square inch pressure. Actually, when the sterilization was evolved, that time Bacillus anthracis pores were used. So several different pressures were created, like 10 pounds per square inch, 12 pounds per square inch, and finally, at that 15 pounds per square inch, where temperature reaches at 121.7 degrees centigrade, it was observed that it is totally sporicidal. So it can destroy even a single spore. So that is why we use 15 pounds per square inch pressure and 121.7 degrees centigrade, not in the other pressure or other temperature. So I must say that the moist heat sterilizing conditions, that is good for the endospore killing, but for food preservation, if we want to make it endospore free, gamma irradiation or cold sterilization is applied to avoid the heat.
Okay, sir. Shayoni Ghosh is asking, is there any bad effect of blanching? I mean, blanching of vegetables with respect to their nutritional properties and all?
I have
It's a repeat
answered.
question, I
Yeah,
think.
I have
Yes,
already
it's a
answered
repeat
on that.
question, Shonima.
Yeah. I think yes.
That has already answered to this question.
Yes.
Yes.
Okay, I think no more questions are there. Okay, there is another question from Dr. Gargi Shahakesh. What is your opinion about getting food from restaurants in this situation?
Uh, you
Sir has
can
already.
you can buy food material from the restaurant, or it depends upon the hygienic condition of the restaurant. Actually, the people who are working in that restaurant, and whether they are wearing gloves or mask or not, because the social distancing and mask, that is the most important thing, what I believe. You can get your restaurant, the food material from the restaurant, you can have that material, but social distancing and the mask, that is must.
Yes, sir. A more or less similar question from Saurabh Bose. He is asking whether it is safe to consume sweets under the present scenario?
I am consuming sweets under this present situation, and just if you believe that the from the shop from where you are actually taking the sweets, that hygienically they are good, you can buy sweets. If it is not hygienically good, then don't buy from that shop.
Okay, thank you, sir. Next question is from Dimpi Thakuria. Is there any negative impact of radiation treatment on foods?
Yes, radiation treatment of food, sometimes it can generate free radicals. So free radicals may mediated oxidized food materials like the protein oxidation or lipid oxidation, certain materials, sometimes that can cause harm. So that is why every time, but I am saying, what I am saying, that it is not that you have to buy the canned material every day. You have to use canned material every day. So if you use it once in a month, it doesn't cause much harm. But if you use regularly that type of material, definitely it can cause harm.
Thank you, sir. Thank you so much. So no more question is there. So thank you once again, sir.
Thank you,
Over to
Sanjay
you.
da.
Thank you. Thank you,
For your,
Mr.
I think I am sure that not only the students, the teachers are also benefited out of your lecture.
Thank
Okay.
you. Thank you.
So you are very patient because you have very patiently handled all the question answers, because we find every time we find you how efficiently you can manage the BOS meeting and all that. So thank you once again, Sanjay
It's
for being with
thank you.
us. Okay, Shuchishmita, now you carry
Yes.
on. Yeah.
Thank you, Professor Ghosh, for being with us for this wonderful, informative presentation and for answering all the queries of the students and even of the teachers so patiently. Thank you, Professor Ghosh. Thank you, Dr. Chatterjee. I know you. I saw you. Yes, you are there for being with us in this session. We'll be waiting for you to hear from you tomorrow. Thank you, Principal Madam, for giving. Yes, sir, you're there. I know. Thank you, Principal Madam. Yes, sir.
Okay.
Yes. Please continue.
Yeah, thank you for such a nice arrangement. Yes, it was a great lecture and we are all benefited by this. And yes, I am also, of course, excited about tomorrow's program. I'm waiting for that patiently.
Thank you. Thank you, Dr. Chatterjee. Thank you, Principal Madam, for giving us this opportunity to organize this webinar. Special thanks to the technical support team, Professor Rituparna Kundu, Professor Pooja Pal, and especially to Dr. Saurav Bose for helping with us with the live YouTube streaming. Thank you, Saurav. Thank you all dear participants for your presence. The feedback form has been provided. Please submit today by 11 p.m. and we all look forward to your presence tomorrow, the last session. Tomorrow we'll have it from 11:30 a.m. Take care, all of you, and have a good day and stay safe. Thank you all of you.
Thank you all. Thank you.
Thank you, sir. Thank you, sir.